So this next section is going to be about sensation and perception. And so we're going to look at each of these. First, we're going to take sensation, and then we're going to look at perception. So let's start with sensation. Or actually, let's look at some key terms that we need to know. Sensation is a process by which our sensory receptors and nervous system receive and represent stimulus energy. Basically, sensation is the ability of our sensory organs to pick up energy in the environment around you and to transmit it to your brain. You know, this happens so quickly and automatically, you might actually take it for granted. To better appreciate your body and brain, think about how your phone constantly is sensing its surrounding. It senses whether an internal signal like Wi-Fi is present, and if so, how strong it is. It senses whether a cellular signal for phone calls and texts is present, and if so, how strong is it? It senses whether a Bluetooth device is in range to connect. It even senses whether a power cord is plugged in into its charging port or if it needs to be charged. Your body senses work in very much the same way. Your eyes, ears, nose, tongue, and skin are constantly monitoring their environment, sending simultaneous messages to your brain about what they detect around you. Perception, on the other hand, is your brain's process of organization and interpreting that sensory information, enabling us to recognize meaningful objects and events. So perception is the ability of your brain to interpret the raw sensations it's taken in. The perception is your brain's ability to translate the sights, sounds, and smells into something meaningful and understanding for yourself. So for example, on the 4th of July, you might see colorful explosions around the sky and hear thunderous booms going on all along with them. Those sights and sounds are initially taken in through your eyes and ears as simple sensation, sheer brightness and loudness. Those sensations become perceptions when you start comprehending what they are. Rather than isolated or unfamiliar sights and sounds, perceptions are things you recognize and put into context. You might see one firework send streams of light cascading down in a circle of curves and say, "Ooh, that one looks like a willow tree." But you might see a bunch of quick flashes light up in one spot in the sky in rapid succession, each producing a single blast of light and sound and say, "Hey, that's cool. It's like a strobe light." Each statement requires that you perceive what those lights and sounds are and what they mean. That is the difference between sensation and perception. The last term we're going to look at is called transduction. It's an essential step in sensation. It's the conversion of energy from outside your body, like light or sound, into neutral energy, like brain activity. Transduction explains how that yellow banana on the table becomes a yellow banana-shaped image in your brain. Transduction explains how the blaring horn of an oncoming truck becomes a blaring horn-like sound in your brain. So it is basically taking the outside stimulus and putting it into a neural signal. So even though your sensation and perception abilities are highly evolved, they do have limits. And so that's what we're going to talk about, are these two different kinds of thresholds. We're actually going to start with absolute threshold. Here it is defined as the lowest level of stimulation that a person can consciously detect 50% of the time the stimulation is present. For example, for one of your senses to notice something, there's to be enough of that something to detect. In other words, the amount has to exceed the absolute threshold, or that minimal level of stimulus necessary for you to detect its presence at least half the time. So, in a silent room, you would hear a dog barking, but could you hear it breathing? On a pitch black night, you would see the brake lights of a car 100 feet in front of you, but could you see them if they were a mile ahead? If you were drinking water, you'd certainly notice if someone had dropped a spoonful of salt into it, but would you notice a single granule? All of these are questions of absolute threshold. Now, you might expect psychologists to have determined the specific measure of absolute threshold for each of your senses. However, these numbers vary so broadly from person to person and even from situation to situation for the same person. That pinpointing any particular amount would be really an oversimplification. The next one is called the just noticeable difference, or some people say the difference threshold. Absolute threshold revolves around a common question regarding the sensation, is it there? That question involves, another common question occurs when you know it's there, but you wonder, is it different? That question involves just noticeable difference, which is the smallest change in a stimulus necessary for you to detect that change at least 50% of the time. What differences are well above the threshold, like the differences between high definition TV and standard definition TV? Now, these are easy to notice, and when the differences are well below this threshold, like the microscopic difference in softness between two Kleenexes from the same box, you don't have a chance in that one. The most interesting difference threshold questions are the ones very close to the threshold itself. For example, imagine that your flashlight is gradually getting dimmer over the course of several hours as its battery runs down. How much brightness will the flashlight have to lose before your eyes notice? Or maybe your old freezer has started to wear out. How many degrees of temperature have to rise before you notice a difference in the coldness of your ice cream on your tongue? So those are the absolute threshold and just noticeable difference thresholds. We're actually going to now move to our first sensation, which is vision. So vision is your sense of sight, and it really dominates the human senses. I mean, all of the senses are important, of course, but we're influenced by what we see more often and more powerfully than by what we hear, smell, taste, or touch. This dominance shows in many ways. For instance, vision occupies more brain space than any other sense. In an online poll asking, hey, if you had to give up a sense, which would it be? Vision ranked last among the 4,000 plus respondents. Only 4% chose it. Even our language reflects how our visual sense permeates the way we think. We use visual terms as metaphors for brain activities that actually have nothing to do with vision. For example, when we understand something, we say, oh, I see what you mean. When we ignore something, we turn a blind eye. When we expect something to happen, we see the writing on the wall. And when we concentrate exclusively on one goal, we have tunnel vision. Now, the hardware of your eye is an engineering marvel. So let's go through the eye as a ray of light does, starting at the outer surface and moving towards the back where the pathway to the brain begins. So light energy is necessary for vision to occur. So here's the eye. We're going to start with your cornea. First, the light hits your cornea, and it's a thin, transparent cover for the whole eye. The cornea refracts images to improve vision, but it cannot change its shape to do so. The iris is just behind the cornea, and it's a colored circular muscle situated in the center of the eye. Now, this iris gives you your eye's unique shade, whether you're blue-eyed or brown-eyed or green-eyed. Next is the lens. After the light travels through the pupil, it moves onto the lens, a clear layer beneath the surface of your eye that maintains focus on the objects by varying its own specific shape. Specifically, the lens changes its own curvature, the degree and shape of the curve, to maximize focus. This is quite similar to the lenses you might choose for your glasses or your camera. You might want the curve to be just right to produce the clearest, most focused image. Unlike the immobile lenses in your glasses or camera, though, your eye's lens can adjust in shapes as necessary, shifting focus between near and far. This is called visual accommodation. It's the process by which the lens changes shape to focus on objects at varying distances from your eyes. Now, as you age, the lens loses some of its elasticity, which is one reason contacts, glasses, or surgery is more likely as you grow older. Once past the lens, light now reaches the retina, which is the rear part of the eyeball that receives visual stimulation and sends it to the brain via the optic nerve. The retina is basically a screen at the back of your eye. The collective goal of all these parts of your eye is to cast a focused, clear image on that screen. That image includes miniature versions of all the items in your field of vision. For example, a single letter in the newspaper headline held at arm's length makes an image about 1.5 millimeter high in your retina. The retina is where visual transduction starts to happen, which means that outside stimuli get converted into internal brain signals. The passageway from the retina to your brain is the optic nerve. Visual information carried by the optic nerve makes its first stop at your thalamus, then moves farther back to your occipital lobe. We also have what's called a blind spot. Also within the retina, it's the part of the retina that contains no rods or cones, which means it can't sense any light. The blind spot is basically the part of the retina where the optic nerve connects. Fortunately, with two eyes focusing on an object from slightly different angles, what strikes the blind spot in one of your eyes will probably be seen normally by the other eye. We also have the fovea, which occupies a small part of the retina. It's an area in the center of the retina that contains many cones but no rods. The fovea, by the way, this comes from the Latin root, that meaning small pit, is where cones are mostly densely packed. So that's where a part of the eye sends as much visual information as possible, especially information containing color and detailed features to the brain. The fovea in the eye of an eagle is even more densely packed than that of humans, which allows eagles to spot prey more than a mile away. Now we're getting to the good stuff, the rods and the cones. The retina contains receptor cells called rods and cones that are specifically designed to detect certain kinds of visual stimuli under certain conditions. So rods are receptor cells in the retina that detect shades of gray and allow us to see in really low light or twilight. Cones are the receptor cells in the retina that detect color when light is plentiful. In the human eye, there are a staggering number of both types of receptor cells in the retina, but far more rods, about 100 million, than cones, which are only about 6 million. And here's a picture of rods and cones. This brings us to the visual information processing. Optic nerves connect to the thalamus in the middle of the brain, and the thalamus then connects to the visual cortex, which is in your occipital lobe. If you'll notice, they cross over though. We talked about this when we were talking about biology, that the right side of your brain, it controls the left side of your body, and the left side of your brain controls the right side of your body. Well, the same thing with your vision. Your right eye is actually connected to the left side of your brain. Now, why do we see the colors we see? That's always kind of an interesting question, and there are several different theories about this. The first one we're going to cover is called the trichromatic theory. So, we discussed the fact that cones are specialized receptors in your retina that detect color, right? Well, the trichromatic theory says that there are actual 3 types of cones, each capable of sensing only a certain range of colors. So first short wavelength sensors, they pick up bluish colors. The middle wavelength sensors, they pick up greenish colors. And the long wavelength sensitive cones, well, they pick up reddish colors. The presence of these three distinct types of cones is the basis of the trichromatic theory of color vision. It's an explanation of color vision based on the idea that your cones are specialized to sense either red, green, or blue. In fact, many TV screens and computer monitors work the same way, portraying a full range of color by mixing just a few. Deficiencies can occur in any one of these three kinds of cones, resulting in distinct kinds of color blindness. Color blindness can be determined with the use of the Ishihara test. If you're looking at the circle below or next to this, you should be seeing a number in the middle. The number is 74. Those people who have color blindness, though, however, cannot see that number. So what do colors look like to a dichromat? So somebody who is colorblind. We're provided some insight by a rare person who was a deuteronope in her left eye, but color normal in her right eye. So what she reported was red, orange, yellow, and green all appeared to have the same yellow hue. Blue-violet appeared to be blue, and blue-green appeared to be neutral. I don't know if you've ever watched any of the videos of people who have received the glasses that help color blindness, but it is amazing and very emotional for them because they never have seen the brightness of green. If they were to look out, think about in Modesto when it is the middle of summer and grass and different things are dead. Maybe looking towards the foothills and you just see a lot of hues of brown. Well, that a lot of time is what a colorblind person or a person who has color blindness, that is kind of what they see the world as. They don't see green grass like the rest of us do. And so when they put on these glasses, suddenly they can see it. Another theory is called the opponent process theory. A guy by the name of Ewald Hearing proposed that we focus 4 primary colors combined in pairs of red, green, blue, yellow, and black, white. To test this theory, I want you to stare at the black dot in the middle of the flag for about 60 seconds. Go ahead and stare, and I will time it for you. Okay, so keep staring. Are you still looking at the black dot? Keep looking at the black dot. Go ahead and allow your eyes to relax while you look at the black dot. Keep looking. I know that you're thinking, this is getting boring, but what do you see now? You should see the British flag. Again, this supports the opponent color theory. Next, we're going to look at hearing. So the word that we use for hearing is actually audition, and it's not just what you do to be on American Idol. That's supposed to be funny if you were like missing out on that. But audition, again, is the sense of hearing. And there are 2 main terms that I want you to know for this, and that is frequency and pitch. Frequency is the number of complete wavelengths that pass a point in a given time. And again, it takes sound energy for you to hear something. And pitch is a tone's highness or lowness depending upon the frequency. Okay, let's talk about the parts of the ear. We are going to divide these into 3 parts. You do not have to know all these technical terms, but I'm just going to go through each part of the ear. We're going to start with the outer ear, which is the part that you're probably most familiar with. The outer ear has 2 parts, the pinna and what we call the tympanic membrane. You have the pinna is the part that you see, right? It is the part of the ear that you can easily touch with your fingers. Your pinna function as a funnel that guides sound towards the inner ear, where the real work of hearing is done. That tympanic membrane is actually, though, in your middle ear. It's right here, okay? So you might also know that as the eardrum. If you ever had, or if you knew somebody who had to have tubes put in their ear, that's where it occurred, was in the tympanic membrane. And tubes are literally, it's a little tiny, tiny tube that they put a little slit in and they place it in there. Interestingly, if a child, and this is who usually gets tubes in their ears, are children and very young children, it's because there's been so many ear infections that this becomes so inflamed that it does not allow fluid to drain out. Believe it or not, your ear is draining fluid quite a lot. That's why that waxy buildup that you get, that's actually a form of the fluid that is coming out. When this gets too inflamed for that fluid to drain, it collects back here and becomes very jelly-like, which can definitely affect a child's hearing. It makes it sound like everybody is underwater. This middle ear also contains 3 tiny bones: the hammer, the anvil, and the stirrup. Of course, this is your stirrup. This is, I believe, the anvil, and this is the hammer, okay? Within the inner ear, we have the cochlea, which is a coiled, bony, fluid-filled tube in the inner ear that transforms sound vibrations into auditory signals. So, one thing, it's really hard to see in this picture, but you might be able to see along in the middle here, there are a bunch of hair cells. One thing that happens is as we get older, those hair cells become brittle. Or if you listen to very loud music, those hair cells can actually break off. Have you ever heard a really high pitch that's just, it's a ringing in your ear? That's usually because those hair cells have been damaged in some way. There's actually a condition called tinnitus that occurs when those have been injured in some way, and that high pitch sound occurs. Now, why do you hear what you hear? There are again a couple of theories regarding this. We have the place theory and the frequency theory. The place theory basically says that different hairs vibrate in the cochlea when they hear different pitches. So some hairs vibrate when they hear high pitches, while others vibrate when they hear low pitches. Frequency theory states that the rate of nerve impulses traveling up the auditory nerve matches the frequency of tone, thus enabling us to sense its pitch. I'm going to try to say that in a little different way. So the explanation of pitch perception based on the idea that a person hears different pitches because nerve impulses travel with different frequencies through the auditory nerve. So they're kind of playing a game of puzzle and match, okay? So that is frequency theory. Now, there are 2 different kinds of hearing impairment, and that's where we're going to finish up today. We're going to talk about conduction deafness and nerve deafness. Conduction deafness is where there's a hearing loss caused by damage to the mechanical system that conducts sound waves to the cochlea. So that, again, you can break your ear or something goes wrong in that process. Nerve damage, it causes nerve deafness. So it's hearing loss caused by damage to the receptor cells or the auditory nerve itself. A lot of times, children that are born with hearing impairments have actual nerve deafness. You may have heard of a cochlear implant. Cochlear implants are actually used to help. They're basically used, they're a small prosthetic device that are worn on both the inside and outside of the ear, and it basically helps to be a conductor for instead of the nerve. That is it for auditory and vision. The next one we're going to look at is going to be the taste and smell, so I'll see you there.